Today we have medicines to treat pain, infection, allergies, high blood pressure, high cholesterol, diabetes and many other problems. But although these medicines benefit many of us, others suffer side effects which can be worse than the disease itself. Ironically, side effects of these drugs designed to help us are now one of our leading causes of death. There is an urgent need to improve current methods of assessing drug safety, both during human trials and before the drugs ever reach the human testing phase. Currently, the linchpin of these pre-clinical methods consists of a series of tests on animals. These are required by the government for the purpose of protecting volunteers in clinical trials, which are the real acid test for all new drugs. Animal testing is usually considered controversial because of the suffering of the animals involved. But many scientists believe that its consequences for human health are equally controversial and deserve more attention. Safer Medicines Trust is a charity whose goal is to improve human health and protect patients by ensuring that all medical research is relevant to humans. Their scientists have studied the available evidence to see whether testing drugs on animals is a reliable way to make sure our medicines are safe. Dr. Jared Bailey is a research associate at the University of Newcastle upon Tyne and consultant to Safer Medicines Trust.
The past 40 years since the UK Medicines Act of 1968, the government has insisted that all new medicines should be tested on two species of animal, one rodent, such as mice or rats, and one non-rodent, such as dogs or monkeys, before they're tested in people for the first time in clinical trials. The Medicines Act was brought in as a response to a terrible tragedy that had been unfolding over the previous 10 years, and which was then the biggest drug disaster the world had ever witnessed. The name of the drug was thalidomide, and from 1957 to 1961 it was given to pregnant women in many countries to treat them for morning sickness. It was heavily promoted as a wonder drug and said to be completely safe, because in many tests in a wide variety of animals it had caused no ill effects, even at huge doses. But in humans, it wasn't safe, particularly in pregnant women. It caused over 10,000 babies to be born with short arms or legs, or with no arms or legs at all.
After more and more children started to be born like me, thalidomide was tested on many different species of animals, almost none of which suffered the side effects shown in humans. Eventually, researchers discovered that New Zealand white rabbits also gave birth to malformed offspring, but only when enormous doses of the drug were used. The bottom line is this: more animal testing would not have prevented the release of thalidomide, because the scientists would not have found the side effects. Even if New Zealand white rabbits had been tested, thalidomide would still have come to market, because a vast majority of the species showed no ill effect from the drug. It amazes me that some people claim that more animal tests would have averted the disaster. The highly respected Office of Health Economics concluded that it is unlikely that specific testing in pregnant animals would have given the necessary warning. The right species would probably never have been used. Furthermore, Dr. John Shardine, an expert in birth defects, commented that in over 50 strains and species in which thalidomide has been tested, including even armadillos, birth defects have only been induced occasionally. So basically, if scientists were seeking to approve thalidomide as a new drug today, the weight of evidence from animal tests would indicate that it should be safe to be used in humans. The thalidomide tragedy happened over 40 years ago. But what have we learned since then? Has science moved on? Can we be sure that a similar tragedy won't happen again? I think that there should be tests to identify such dangerous drugs and chemicals. But which tests are up to the job?
I recently reviewed the success of tests that have been carried out for over 40 years since thalidomide to predict the potential of drugs and chemicals to cause birth defects in people. The results show that these tests, using animals as diverse as rats, dogs, cows and monkeys, agree with results in humans only around half of the time. We might as well have flipped a coin. I also reviewed studies that used in vitro or test tube-based methods and found that these are significantly better at predicting the response in humans, with up to 80% accuracy. So we do have superior methods, but our regulations are stuck in the past. The crucial question is, can we be sure that there will never be another thalidomide? Well, let's look at what happened recently with the drug Vioxx. Vioxx is a painkiller that was prescribed mainly for arthritis between the year 2000 and 2004. It was tested in animals and found to be good for the heart, but it went on to cause over 300,000 heart attacks and strokes in people, nearly half of which were fatal. This is the biggest drug disaster in history, many times bigger than thalidomide.
It's clear that there are serious problems with our current systems for making sure new medicines are safe and effective. For example, in March 2006, a clinical trial at London's Northwick Park Hospital made headlines around the world when 6 healthy young men were nearly killed by a new drug which was given to them after it had been shown to be safe in monkeys at 500 times the dose. The US Food and Drug Administration, the world's largest drug regulator, has complained that we're using 20th century tools to develop 21st century medicines. Fortunately, there are many new technologies and companies addressing this problem by focusing on the way that the human body responds to new drugs before actually testing them out on people in phase 1, 2 and 3 clinical trials. Randall Charlton is the founder of Asterand, the world's leading human tissue company.
I think it's self-evident that mice are not men, and in order to develop drugs with more confidence, wherever possible, one should use the human model, the species in which the drug is eventually going to be given. We operate a network of hospitals around the world in about 15 countries, and we collect under strict ethical conditions tiny amounts of human tissue backed by large amounts of data, all properly anonymized to protect patient and donor identity. Perhaps I should introduce again my colleague Bob Coleman, who has had more experience than pretty much any other scientist I can think of in working with human tissue.
It's important to remember that the pharmaceutical industry is developing drugs for use in humans. And for that reason, a focus on human biology, I believe, is very important. Now, historically, the industry has relied very much on the use of animals, both isolated tissues and whole animals. But it's also had its problems. I've got many years' experience in the industry, and I worked for 30 years in a major pharmaceutical company where the use of animal tissues was the norm. And the thing that frustrated me there was the lack of predictivity of many of the data that we generated. And even at that time, I made as much use as I could of human tissue in trying to focus on what I thought was relevant. There are many needs for improved medicine in the world today. There are diseases of the lung, of the bowel, of the cardiovascular system, of the brain, all of which need improved treatments. The industry is finding it increasingly difficult to get new drugs onto the market. The time taken is colossal. It takes something like 15 years to get a drug to market, and the cost is estimated to be of the order of $1.6 billion per drug. And even when they get onto the market, drugs aren't necessarily totally trouble-free. The role of Asterand is to provide high-quality tissues and high-quality services to enable the pharmaceutical industry to more effectively get safe and effective drugs to the market.
An ingenious new technology that brings us closer to mimicking the whole human body in the safety of the laboratory is being pioneered by Hurel Corporation in California. Dr. Greg Baxter is the co-founder of Hurel.
Accurate prediction of human response is difficult, often unreliable, and invariably expensive. There is considerable concern whether animal studies can predict human risk precisely enough, and traditional cell culture assays do not accurately mimic the complex environment a drug encounters within the human body. To address these limitations, we have developed an interactive, cell-based microfluidic biochip. The Hurel, which is a condensation of the words human and relevant, is composed of a network of interconnected microfluidic compartments and channels, which replicate the organ and the blood system of the human body. Each compartment represents a different organ or tissue. For example, an early prototype chip was designed to investigate inhaled toxicants, such as naphthalene. In this example, there is a lung compartment as a target tissue for toxicity, a liver compartment, which is important for drug metabolism, a fat compartment, which is important for accumulating drugs and breakdown products, and an other tissues compartment, which replicates the holdup of circulating blood in other tissues of the body. The culture medium is a blood surrogate and recirculates in the Hurel system just as blood recirculates in the human body. The prototype biochips are roughly the size of a postage stamp and are fabricated in silicon using standard microfabrication techniques traditionally used in the microelectronics industry. But most importantly, the geometry of the chambers and channels are designed to mimic key aspects of the human body, such as the time taken for a drug to circulate and where it goes in the body. With the Hurel system, drugs are exposed to a more realistic physiological environment, thus providing higher and more accurate informational content than traditional in vitro assays.
Another way to predict what effects a drug may have on the human body is to find out which genes it affects and in what way. This can be done using DNA chips, small glass slides the size of a postage stamp containing thousands of tiny wells. Samples of DNA are placed in these wells and a computer detects which genes are being activated or suppressed by the drug in question. A Cambridge company, Simugen, uses the latest cell and DNA-based methods combined with advanced computer programs to predict how toxic a drug will be. Their methods have been able to detect hazards to the human liver that were missed by animal tests. Dr. Quinn Wills is the co-founder and chief scientific officer of Simugen.
The question is, how does one predict the toxicity of a molecule before we put it into human beings? And we believe as a company the only way to really get this right is to look under the hood of a cell. And by that I mean have a look at what the genes are doing. So what we can do is allow you to culture chemicals with human cells, have a look at the genes, and then actually put that into models of how we think this translates into toxicity. Doing that, we can tell you what kind of toxicity we think is going to come up, at what dose do we think this toxicity is going to come, very important, and we can give you some feel for what you could be doing to your molecule or your drug or whatever it is you're creating to make it less toxic. What we do is use mathematical models of how the genes are actually changing to predict those dose-response relationships. So it's a very, very exciting technology because we're addressing several things. First of all, we're giving you this richness of information that current methods can't give you. We can predict toxicity, we can tell you the level at which it's going to be toxic, we can give you some indication of what you could be doing to make it less toxic, and we're doing it in a way that is very cheap, very fast. It takes 24 hours to do a cell culture. So this is something you can use right at the beginning of, for example, a drug development pipeline as a company, which will save an enormous amount of money and decrease the reliance on animal testing.
A further way of predicting what effects a new drug may have on the human body is by computer modelling. Professor Dennis Noble of Oxford University was the first scientist to generate a computer-based model of beating heart cells in 1960. Although all of his early work was based on information gained from animal hearts, with his collaborators, he has now pioneered the development of models based on the human heart.
The image you're looking at may appear to be a film of a living human heart, but it isn't. It's a highly sophisticated computer program using tens of millions of separate equations, combining graphics and mathematics to create one of the first of a new generation of simulations called virtual organs. These are set to become some of the most powerful tools we have in the fight against disease, with scores of uses from disease diagnosis and drug design to training and research.
My involvement in modelling the heart and other organs began over 46 years ago when I was working in London. I was the first person to build a computer model of the activity of individual heart cells as they were beating. And that was based on experimental information that we obtained on the nature of the processes occurring in those cardiac cells. Now, those models were very simple and they ran on very, very simple computers. Nowadays, 40 odd years later, we have masses of more information, thousands of laboratories all over the world accumulating experimental information. The models have become very much more sophisticated, and most importantly, they've been incorporated into models of the whole organ, so that you can actually see the organ itself beating away.
The virtual heart allows doctors to study the effects of a heart attack in controlled circumstances without endangering a real patient. And unlike real life, in which a heart attack can take just a few seconds, the progress of the attack in the virtual heart can be slowed down, speeded up, and repeated at will, enabling scientists to measure exactly what is happening.
They've become sufficiently sophisticated that they are now used in pharmaceutical research and for assessment of drug action by regulatory agencies. 10 years ago, or nearly 10 years ago, I was involved in using one of these models to help a major pharmaceutical company, Roche, to process the drug application that they were making to the Food and Drug Administration in Washington at that time, and we successfully helped them to get their application through. And there have been other examples more recently where companies like Novartis have used the model to clarify the actions of the drugs that they are developing. So the work is already partially achieving the object of enabling some of the work that is normally done on animals to be done instead on computer simulations. But I should emphasize this is early days. We can't yet look to a time when you could replace animal experimentation entirely by computer simulation. But clearly the long-term goal would be to achieve a situation where, just as in the aircraft industry, you don't build an aircraft before simulating it on a computer, in the future people will not build new compounds, new drugs, new medical devices without simulating their actions on the various organs of the body.
Virtual organs such as this one allow researchers to develop and test new drugs faster, more confidently, and at less cost.
One of the best ways of finding out how a new drug will behave in the human body is to give it to volunteers at a minute, safe dose. Then, by using incredibly sensitive measuring devices, scientists can monitor where the drug goes and how it is broken down in the human system. Microdosing is an exciting new breakthrough in drug testing. It uses machines so sensitive that they could detect a litre of liquid after it has been diluted in all the oceans of the world. Acceleron is the world's leading microdosing company, of which Professor Colin Garner is the founder and chief executive officer.
Now, why is microdosing of interest? Well, it's of interest because it allows us to get human metabolism information much earlier, much more safely, and with minimal use of animals. Now, the great attraction of microdosing from a drug development point of view is that you can take your drugs into humans with much reduced costs and much reduced time compared to classical, traditional so-called phase 1 methods. So typically, to take a molecule into man using classical phase 1 methods might cost as much as £3 to £5 million and take anywhere between 12 and 18 months. Whereas using the microdosing approach, we can do the same type of study at much less cost, typically around about £250,000, and get the results in 4 to 6 months. So there is a time and cost saving to the approach, and it's been estimated that by 2008, as many as 40% of pharmaceutical and biotechnology companies around the world will be using microdosing. 2010, nearly 90% of all companies have stated that they will be using microdosing routinely in their drug development program.
These new technologies enable drugs to be developed and tested specifically for humans. Yet there is still a vocal strand of opinion which insists that new treatments will only come from research involving animals, and that it would be unethical to expose people to drugs without first showing that they are safe in animals. But this view is increasingly questioned by members of the medical profession. Kathy Archibald is the director of Safer Medicines Trust, who commissioned a survey of family doctors.
We commissioned one of the world's largest market research companies to conduct a survey of 500 GPs across the UK. The survey found that 83% of GPs share our desire to see the medical value of animal tests evaluated scientifically. That's an extraordinary level of concern. I know from my own experience as a scientist working in drug development and as a patient with a serious illness that much animal research is simply irrelevant to human medicine. What happens in animals doesn't matter. It's what happens in people that counts. My disease was in the pancreas, which scientists have just discovered is so dramatically different between rodents and humans that studying mice and rats is futile. The object of study has to be human. We acknowledge that there's no such thing as a drug that will be absolutely safe for everybody, but we do believe that drugs could and should be safer than Vioxx, for example. And we believe that one reason why clinical trials are risky is that very often, instead of information about safety in humans, all we have is an assurance of safety in animals, which, as the story of thalidomide should have taught us, means very little. The fact that 9 out of 10 drugs fail in clinical trials proves that point very clearly. So does the fact that prescription medicines are our 4th biggest killer after cancer, heart disease and stroke. We believe that drug trial volunteers should be protected from risk by making sure before clinical trials that the new drug is as safe as possible, something that may well best be established by a battery of human-based tests such as those we've just heard about.
We've heard from the experts about the potential of the latest methods for assessing new drugs. But are the people responsible for legislating for drug safety listening? Many politicians are concerned about animal experimentation, but believe there is a dilemma between protecting animal welfare and advancing medicine. Yet there is growing evidence that favouring the state-of-the-art methods we've seen in this film would benefit medical progress as well as laboratory animals. Tony Benn was a Member of Parliament for over 50 years and a Cabinet Minister for 15 of those. He's given the matter some serious consideration.
A lot of doctors and scientists are now challenging the scientific value of the use of animal testing for human purposes. And these arguments have to be taken seriously. 250 members of Parliament have signed a motion in the House of Commons calling for a government inquiry. And that call has the support of a lot of people from the medical profession and, of course, from the public as well. And what is really required is a proper scientific evaluation of the methods that are used to decide, or make it possible for us to decide, what is the value of animal testing. And it's never been done before, and there's nothing whatever to hide, so I can't see any case against it. This is not an argument against science, quite the opposite. It's an argument for bringing modern scientific thinking to bear on an issue that has in the past been simply described as if it was those who in favour of animal rights against scientists. It is actually a very important scientific development. And all that we want to see is the government to set up a review where these arguments can be properly evaluated in a scientific way, because quite apart from anything else, thousands of people suffer from the side effects of drugs, and some of those ill effects might be averted by using better methods. And the government should now, in my opinion, make this a priority, because if they did that, then this argument could be settled in the best possible way for the benefit of everybody concerned.
Safer Medicines Trust believes the government should commission an independent scientific comparison of the results from animal tests with those from a battery of human-based tests to see which more accurately predict the human response. Most MPs and GPs support this idea. The outcome couldn't be more important for those of us who await new drugs for ourselves or our loved ones, or for the volunteers who take part in clinical trials for the benefit of society and medical progress. We hope that this short film has offered a glimpse of the many new techniques that are paving the way to safer medicines for us all, thanks to their focus on human biology.